Hi everyone, my name is Tiffany. My teammates are Vian and Thang. Together we present to you a DNA assembly comparison tool. Our tool takes into consideration how sequence attributes affect assembly. For example, high GC content cause the formation of stable secondary structures which are difficult to unwind during assembly. Restriction sites that are present within the sequence to be assembled also interferes with restriction enzymes used. Melting temperature also plays a part, as high temperature can cause misannealing or mispriming of template and complementary strand. The number of strands to assemble also affects the process, as double-stranded DNA, for example, is more expensive to assemble than single-stranded DNA due to the increase in time and material required. The presence of repetitive motifs may also cause assembly complications for certain methods. As we go through this video, we will show you how our processing algorithm checks these attributes when determining the best company and best assembly method for the sequence. Let's click Assemble My Sequence to begin. First, I'll demonstrate how our front end performs error checking, where if a user inputs anything other than a valid sequence, it will output an error message and all of the resulting variables become undefined. So for the first example, we will use a random DNA generator to test out a DNA sequence of 600 base pairs in length and a GC content of 40%. So click Generate, and we'll copy paste this and input it in the textbook. And then we will select double-stranded DNA and press Submit. As you can see here in the results box, our recommendation of the best company and the best assembly method is displayed, along with other details such as the price, the sequence length, the turnaround time to assemble the sequence, the GC content, and the sequence fold score. If the user wants to view the results from other companies in the database, they can simply click this Company Details button, which will bring them to this page containing company cards, where each card contains information about each company. When the user hovers over the company card, they can see the company's capabilities and requirements, which affected the final pricing of the sequence. This will be further explained by Thang when he goes over the database models. And if you go back and click on this assembly method details button, you can see how the sequence was split into parts based on which assembly method was used. The page displays similar cards, but now displaying more information about each assembly method, and if you hover over the card, you can see the parts that this sequence was split into. If you're wondering why BioBrick has a turnaround time of 0 and cost of 0, it's likely that the sequence could not be assembled with BioBricks. And here you can see that for Golden Gate, the sequence was split into small parts, but for Gibson, it was not. Clicking the Home button brings the user back to the main page where they can then assemble a new sequence. So now I will demonstrate how our tool can also handle inputs in the form of amino acid, one-letter representation of the codon. Next, select amino acids and click Submit. And similarly, if you click Company Details, you get to see the companies again. And if you click Assembly Method Details, you can also see details about the assembly methods and how the parts were split.
Our API is written in Django in the language of Python, which connects to the MongoDB Atlas database. We utilize the four basic functions of the request, which is get, post, delete, and put. For us, we will grab the company data using one of these requests, which will grab all the company data in a database, while also grabbing the user input sequence and the user input type from the front end. Afterwards, we combine the user input with all the company data into a processing algorithm which can calculate individual company costs, individual assembly costs, and individual turnaround time. And then using that, we will determine the best company and the best assembly method, which will then display on the screen the results sequentially.
Each company in the database will have their company name, the price per base pair length minimum, threshold, and maximum. If the sequence length is over the maximum or under the minimum, then the company will refuse to synthesize a DNA sequence. If it's over the threshold and under the maximum, then there will be a price increase per base pair set to be the blank price increase. The same concept will apply to GC content and homology, which is also fold score. Furthermore, there will be a price increase if the sequence is a double-stranded DNA. Also, we have made it so that certain company can synthesize amino acid sequence or not.
For the request we give to the API, we mostly use like the delete request, which first takes the companies and the user data, and then put both of them into a function called all company, which output the best company and best assembly method with all the costs, and then we return that as a JSON file. The best company data have an all company function which go through each individual companies and roll through another function called price of company, which takes in the sequence object and each individual company and find the actual prices. And we create an object of individual company with the price and add it to a list, and then have a object of parts of all the individual assembly method and also its parts, cost, and time, and also the sequences length, GC content, floating score, best assembly, and turn time. We put this all into a list and return it as a JSON file.
So now we're going to talk about how we split the DNA sequence that is given based on the DNA assembly method. And by doing so, we could then update the cost and the turnaround time for each assembly method. So over here, we have the splitting of the sequence based on the BioBrick assembly method. And to do so, we read from the BioBrick Standard 10 library. Then we check whether or not certain subsequences of the DNA sequence are part of the BioBrick library. Then we check the remaining, the remnants of the DNA sequence, and we check for restriction sites, which would then be illegal in creating a new BioBrick. If it passes that, then we can create new BioBricks and be able to assemble the sequence within the BioBrick standard. We also update cost and turnaround time for the BioBrick based on how long it takes to join each pair of BioBrick parts. We also split the sequence basis based on the Golden Gate assembly method. And to do so, we have to divide the sequence recursively into parts of suitable size, which the Golden Gate method could accommodate. Then we check for conflicting overhang sequences of length 4 for all these different subparts. We penalize the cost and make the method illegal based on whether or not the parts meet the criterion for the melting temperature, as Golden Gate has to cycle between 37 degrees Celsius and 16 degrees Celsius in order to join parts together in the one-pot assembly method. Since Golden Gate is a one-pot assembly method, turnaround time is about the same unless new parts have to be synthesized and certain attributes such as the length of the parts contribute to an increase in that time. Lastly, we have the Gibson assembly method. And to do so, we first divide the DNA sequence into 5 or less parts of suitable size. Then, like Golden Gate, we check for conflicting potential overhangs of about size 40 within the DNA segment, such that assembly can occur without any issues in the order of the DNA sequence being assembled. Lastly, we randomly sample and optimize, and then we penalize the cost based on melting temperature of the DNA parts, as well as the secondary structure of the parts.
To demonstrate that our tool actually considers the sequence attributes when comparing it to the requirements of each assembly method, we will be testing a sequence that was optimized for BioBrick assembly, as it is composed of parts that already exist in the BioBrick library. Thus, as a result, it will produce the lowest cost using BioBrick assembly method. So let's do single-stranded DNA this time and press submit. Just to compare it with the other assembly methods, I will show you the assembly method details page. Here we can see that if the assembly method is BioBricks, the cost is $7.5, while for Gibson it's $8.4, and for Golden Gate it's $53.3. As you can see here, although Golden Gate had a shorter turnaround time, our tool prioritizes lower costs over faster turnaround time.